got a little bit of writing to do just to finish off. So we're saying whenever we're doing field work, we take samples. And the sample should be a representative of the area. Okay? Because we don't have enough time to count all of the wood lice in a forest or all of the daisies in a field. And so we just take samples. Okay? So if you're sampling plants, we use a quadrat. So that's the name of the little frame. Okay? And the ones we use in school have sides that are half a metre, 0.5 metres. And the area, therefore, is a quarter of a square metre. So the area is 0.25 metres squared. You can get bigger ones, you can get smaller ones. And if you got a question, it would always tell you in the question the size of the quadrat that we're using. Okay, we've sort of talked down through this, and we're saying that it's really important that the quadrats are positioned randomly, not just put in one area, one corner. So how do we do this? So you put two tape measures at right angles. And use pairs of random numbers as coordinates. And you put the quadrat down. Okay, so we're just after saying that if you were looking at, I'm just repeating this for the people that aren't here, that page 14, fieldwork page, so the people that weren't here, that's page 14. So percentage cover. So species A, what would you say for species A? 50%. 50%. Yep. Alana, what about B? 15 or so. I would say it's about 10. 15 wasn't a bad guess. About 10%. Species C.
26. 22.
Hold on a wee second. I haven't drawn my lines too well.
29.
20. I'm going for 25%. Okay. So it is an estimate. It's your best guess, really. And generally, you would round it up to the nearest 5% or so. So you're not sort of looking for figures like 29, 28. We're not that accurate. So roughly, is it half? Is it a quarter? Is it 10%, 20%? You're estimating. Reasons why you would do this. You would say, well, first of all, we said it may be difficult to distinguish individual plants. Okay? So, for example, grass. You haven't a clue how many grass plants there are in a field.
What's the word after?
Secondly, sometimes you're looking at so many things. So, I'll show you a few photographs in a minute or two. In fact, maybe if I show you a photograph of barnacles. So we're talking there about barnacles not being able to count them. So you'd say another reason for estimating percentage cover would be large numbers. Would be too time consuming. To count. So, we've worked out how you would take a random sample. Okay? And we talked about if you were doing a survey in Dungannon about whether people preferred something or the other, you're not just going to ask one person. The reason for that is that you want reliable results. Now, what do I mean by reliable? Reliable results, if you do an experiment over and over and over and over again, you're going to get similar results. We say that those results are reliable. Okay? Same way as if I had to go out of the classroom because it was cold out and I came back and Jada is lying on the floor.
Is she living still?
Debatable. Blood coming out of her head, all right? So I am going to say, Gabrielle, what happened? And if Gabrielle says, she was sitting there, she bent over to get something out of her bag, but she hit her head on the corner of the table and she fell, right? And if I say to Avis what happened, and he said, Gabrielle started fighting with her and, punched her, she hit her head and started bleeding, collapsed on the floor. If I say to Jake, and Jake said, no, it was Nikki who was actually having a fight and she hit her over the head with a stool. Have you a clue what actually happened from those reports? No, they're all different. Would you say that those people are reliable witnesses?
Yep. I'm very reliable.
Girls, that's not what I asked. I said, would you say that those people were reliable witnesses whenever they gave three completely different versions of the same thing?
One of them might be.
Yeah, but as a group, are the three of them reliable?
Yeah.
No. If you did an experiment, okay, you did an experiment, so maybe it's a physics experiment, you put a 1 Newton weight on the end of a spring and you work out the extension of the spring and you do it 10 times. And you get figures like 90 millimeters, 90 millimeters, 89 millimeters, 90 millimeters. Are those reliable results? Girls, I am getting very frustrated with some of you because you're not listening. And to make matters worse, you're talking. So let's just cut it out for the rest of this period. Okay, so you've got a small number of figures, but they are telling you the same result, basically. There's very little variation. Okay, we've got one, very slightly different, but it is really close to the other three. So when you do an experiment or when you do a survey and you get the same result or very close results, you say that they are reliable. Now, if you only do the experiment once, you don't know whether the results are reliable or not. And so to improve the reliability, you repeat your readings or repeat the experiment or you take more samples. So if I wanted to find out whether pupils in year 11 preferred Scooby-Doo or SpongeBob SquarePants, I would ask all of you and I might ask a few more people, all right? Shh. If I only ask 2 of you, that's not considered reliable, but to increase the reliability of your results, you take more readings, if it's that kind of physics experiment, or you repeat the experiment more times, or in the case of field work, you take more samples, which means you put more quadrats down in this case. All right? So if we look at something like dandelion plants. Okay, you've got 4 plants in there. It's quite easy to count. And we said that if you've got one that's more outside than in, you wouldn't count it. Whereas if you had one that was on the edge like that, you would count it. So if it's half or more, you count it if it's inside. But we've just said, trying to count grass. If you go out to a field, Zoe, if you go out to a field to count grass plants, it's impossible because you cannot distinguish where one plant starts, another plant stops. So what we would do if you had a quadrat, okay, so say you had that amount of grass, and maybe you had a wee patch of clover, and maybe you had a few patches of buttercups. Zoe, there's a big empty table at the front here, so if I catch you talking again, it's either going to be you or Clara or whoever you're talking. You know, if you're the one that's talking, you're moving, all right? So what we do here is we estimate the percentage cover. All right, so roughly, if you were estimating, if you look at the grass, Sam, how much do you think of that?
60%.
Pardon?
60%.
About 60%, so you'd say grass, 60%, clover.
20.
I'm not sure that there's 20, because if you divided that up into quarters, quarter is 25%. So maybe 5%, between 5 and 10%. So maybe we might just say 5%. Buttercup, 5%. Oh, there's another 5%. So altogether, because you've got 2 there, you might say 10%. You get the idea? Okay, roughly, you saw quadrats yesterday. Roughly, there's the average hand. So if you put your hand like that, the average hand is 5%. So that's a good way of estimating percentage cover in a quadrat. So if we look at the next page of notes, it's saying there, 1 quadrat wouldn't give reliable results. And we've just explained that. To gain reliable results, it's important to take values from a number of quadrats, usually at least 20. So if you can highlight reliable results, and I would just work put repeats in there. So in other words, you repeat an experiment, or in this case, you're taking at least 20 quadrats. So getting back to the scenario where Jade is lying on the floor with blood coming out of her head, if I gave everybody a piece of paper and told you to write down a report of what you saw happening, and everybody gave in a piece of paper that said Jada reached down to her bag to get something but hit her head on the table and collapsed. And if everybody has written down the same version and you've had no time to compare or anything, well then if I'm reading those, I'm going, well, that looks like pretty reliable witnesses. They're all telling me the same thing. So this term, reliable results. Are the results consistent? Are you getting the same pattern? Now, in this case here, you may not be getting the same numbers, but if they're close, you'd say they're pretty reliable. So say you've got all the data from your quadrats and you've worked out, you've got maybe 6 dandelions in this quadrat, you've got 7 in this one. You might have none in another one. There might be quite a lot of variation. What you're going to have to do, though, is you're going to average the number of dandelions or daisies or whatever species it is per quadrat. In some cases, you might use percentage cover, and so again, you'd work out an average again. So, here's an example. So, we've got dandelions in a field. This is the area or the size of our field. And we took quadrats that have a side length of 1/2 metre. So, think back to yesterday, a quadrat, 1/2 metre by 1/2 metre. What area does it cover, Jake? 0.25. So we'll write that in first of all. Now, in this little example, they've only looked at 3 quadrats. Now, that's not very good. But to make things easier for you, you've got 3 sets of figures. What's the average number of dandelions per quadrat there, Elena? 3, yep. So 3 per quadrat. So we've got 3 per quadrat. Each quadrat is 0.25 of a square metre. So if we want to work out how many there are per square metre, we take the average, divide it by the area of the quadrat, and that gives us 12. Dylan, what's the area of our field? What do we have to multiply?
The quadrats by the area.
What's the area of the field, though? If you look over here, it says the field measures 30 by 20. So the area of our field is 30 multiplied by 20 metres. And that should give you 600 metres squared. So what we're saying is we've got a field, there's 600 square metres. We worked out that Girls, what is there to talk about at the moment?
I can't say the average number of dandelions per metre squared.
12, but that was a good while ago. So we said 3 per quadrat, but a quadrat's only 1/4 of a square metre. So that works out at 12 per square metre. We've got 600 square metres, so we need to multiply those together. So it's 12 times 600. And I'm just going to add in there average per metre squared times by the total area. So 12 600ths should work out at, is it 7,200? Yeah? So, average per square metre times by the total area, in this case 7,200, all right? So that is if you had one area that is pretty much the same. So a field or a lawn or an area of grassland. And if you want to estimate the population size, so the number of plants, or you could use things, if you're on a rocky shore, you could look at limpets, you could look at barnacles, very slow animals that don't move very much. It's not going to work for something like insects that can hop out of your quadrat, or field mice or anything like that. It's only used for plants. It's only used for plants or animals that don't move very much. But sometimes we want to actually look, and if there's an area, so say for example, Yeah. So, belt transect, use in habitats where there's gradual change from one side of the habitat to the other. So we were looking at a slope there, but equally you could be looking at sampling. So again, you're going to use a quadrat, so you can highlight that. Somewhere like a rocky shore, you're talking about limpets and barnacles, animals that aren't moving around, and so you could sample them using a quadrat. In a belt transect, quadrats are placed along a line from the bottom of the shore to the top of the shore. And they could be placed continuously, so that's where they're placed one beside the next, or at intervals, so e.g. every 5 metres. So this diagram at the bottom is showing a belt transect. So you've got your first quadrat at 0, your next one is right beside it, and so on the whole way up the shore. The one here, this is called an interrupted belt transect because it's not continuous. Quadrats are going down at a regular interval, maybe every 2 metres or every 5 metres. Okay, homework. Monday is a holiday. I will not set you one this weekend then. At a rocky shore. A rocky shore, we're not talking about a beach. That's a sandy shore. A rocky shore where you've got lots of rocks, you've got rock pools and so on. So if these are your rocks, so it's like a cross section, there's the sea. The sea goes out and the tide goes out and comes back in again. You're going to have areas up at the top here that never get covered by the tide. So you're going to get this area up here where it's going to be really dry. You've got rocks, the waves don't reach it. So the seaweed that lives there is going to be at danger of drying out. So it needs to have special characteristics to stop it from drying out. Anything down here is going to get covered in water for several hours during the day. Anything down here probably going to get covered in waters for 12 hours of the day. Anything down here is going to be submerged in water for nearly all of the day. And so you find down here you've got long seaweeds seaweeds are attached to rocks, but they need light for photosynthesis. So the seaweeds down there tend to be long seaweeds that can float with little air bladders so that they can float and get sunlight. Seaweeds that have grown up here are much smaller surface area, so they don't dry out. And they tend to have very thick waxy layers so they don't lose water by evaporation. So if you wanted to look at, where the different seaweeds were found, and you wanted to work out was there a pattern, you would get tape measure. When the tide goes out, you would put it down at low tide and put it up there. Might go from 0 to 100 metres. In that case, you might put a quadrat down every 5 metres. Okay. So that's called belt. Well, you had there was a stream flowing through an area. And obviously, if we go up the slope from the stream, maybe you find at the top of this hill there were trees growing, a few bushes growing here, grass down here. Basically, we've got a change in the environment. As we go from down here up that slope, we've got a different environment. It's going to be dark under these trees at the top. You're going to get a lot of light here. It's probably going to be very moist. The soil down here, close to the stream or the river, is going to be pretty pretty moist. So where you've got a change in the environment, a gradual change in the environment, you're going to get change in the species that are growing there. So at the top here, we've got trees. At the bottom, we don't have trees. We just seem to have grass. You might have some bushes in the middle there. But you've got differences in light intensity. It might be cooler underneath the trees because you're in the shade. So the temperature might change. You might have moisture content of the soil. It might be drier at the top. Wetter down at the bottom of the slope. Underneath the trees, you might have a lot of nutrients because the leaves are falling off, the nutrients are going into the soil. So you might have different nitrate concentrations, different other sort of ion concentrations. pH of the soil might be different in different places. Those factors, if you think back to early on when we looked at words like environment and habitat and so on, those are environmental factors that are non-living. What was the word that meant non-living factors, like temperature and oxygen content? Abiotic. So those abiotic factors will affect what plants grow there, what animals grow there. Some plants can only grow in areas where the pH of the soil is 7. Some will grow where it's slightly acidic. Some will grow where it's alkaline. And if you dig them up and plant them somewhere else where it's got the wrong pH, they'll not grow. And we talked about animals being adapted. Plants are adapted as well. So if you had somewhere where there was a definite change in those abiotic factors, if the environment changed, you would want to survey that area. But there's no point just going in and randomly putting quadrats down because you're not going to see any patterns because your quadrats are just going to be randomly distributed in the area. And so what you do is actually get a tape measure and put it down along that line. So get your tape measure, put it on the line. That could be 0 meters, that might be 20 meters. And what you could do is put a quadrat down every 2 meters or every meter. Depends how far you're going there. So you'd probably want to put it down every 2 meters, so you'd have 10 quadrats down there. And we call that a transect, taking a transect through the environment. It depends on the length of it. So if it was 20 meters, I would be putting a quadrat down every 2 meters, so I've got 10 readings. If it was a much shorter area, or if you had more time, you could actually put your quadrats down end to end. And so you're actually sampling the whole, like a strip, a whole belt of land. And so that's called a belt transect. Okay, so the key things there, it's used in a habitat where there's a gradual change from one side to the other. So if you go to your notes there,